Hi everybody. Evidently my first attempt to create a screencast failed in the upload, and unfortunately I'm at home and that video is saved on my desktop computer, so I'm going to go back and remake the video for you. So here we go. We ended up with quantitative traits. So we were talking about how quantitative traits are determined by multiple genes that all contribute to a single trait, and the net effect of each of those genes is that each dominant allele adds a little bit to the trait. And so oftentimes these are called additive genetic traits, they're called polygenic traits, they're also called quantitative traits. And we talked about last time that they are quantitative traits because they take on numerical distributions of phenotypic trait value. And so here you can see the dominant number of alleles takes on a normal shaped curve where the average phenotype is at the peak of the curve and the most extreme phenotypes are out here at the tails. So the net effect of these types of traits is that oftentimes we don't necessarily need to know exactly which genes are responsible for the trait. We can just treat these traits as distributions of trait value, and we can analyze them with statistical techniques rather than doing things like Punnett squares and things like that. But remember that the way we got to this quantitative trait distribution was actually by doing a Punnett squared with a trihybrid cross that demonstrated this normal distribution of dominant alleles. So when we start to look at these quantitative traits in terms of thinking about evolution, which is kind of the point of this section of the course, we can have something like this. This is data from a former class of mine where I asked all the students in the class to get the average offspring height of them and their adult siblings. Obviously, young siblings wouldn't work because they would be shorter than their adult height. And then I have on the x-axis down here the mid-parent height, and this line represents the correlation between average offspring height and mid-parent height. And when we look at this, basically, if you are a tall parent, then presumably you have a lot of dominant alleles that make you tall, and those parents would lead to offspring that are also tall. If you're a short parent, you have fewer dominant alleles, and so you're going to give rise to offspring that are shorter. And so what relates the phenotype of the parent to the phenotype of the offspring is the slope of this line. And the slope of the line in a parent-offspring regression, which is what we call this particular formulation of data, the slope of that line is called the heritability. And so if we were to imagine this population and say, well, we're going to prevent the lower half of this distribution of parents from reproducing. So that's what all these red dots are on the figure. What that would do is it would take this pre-selection average of the parents, and that pre-selection average would correspond to a pre-selection expectation of what offspring height would be. But when we prevent those below average height parents from reproducing, that increases the average height of the parental population, and as a result, that would also lead to a concomitant increase in the average offspring height. And the degree to which the parental generation shifts its mean, that's the strength of selection. And the degree to which the expectation of what the offspring would be is referred to as the evolutionary response to selection. So we're going to indicate that with an R on this figure. The S, the little s, is the strength of selection. Now, if there's a lot of selection, then the response to selection will be higher. If there's a little bit of selection, the response to selection will be lower. But in all cases, what mediates the response to selection is this slope of the line and how the slope of the line influences where the pre-selection mean of parents and the post-selection mean of parents intersect that line. And so the slope of that line is heritability. Now the symbol for heritability is h squared. And this equation at the top of this figure shows you the line, the equation for the line 5.686 is the y-intercept, 0.9165 is the slope, and that slope, 0.9165, is the heritability, and heritability is just equal to h squared. You don't have to do anything to that 0.9165. 9165 is just it. This confuses students sometimes because they're like, well, to get heritability, do I have to take the square root of 0.9165? And you don't. H squared equals 0.9165. So get that clear in your head. I can explain where the h squared comes from. This formulation of thinking about quantitative traits was developed by a guy named Ronald Fisher. He was an evolutionary biologist slash geneticist, but he was also a statistician. And when he thought about these types of traits, he basically dubbed heritability as additive genetic variance. It's the variation in the trait that is due to the additive effects of these genes. And so when he thought about heritability, he thought about it as one form of variation. And since sample variance is s squared, he thought that the symbol for heritability should also be squared because it is genetic variance as opposed to being sample variance. But there is no genetic standard deviation, there's just genetic variance. And so genetic variance is h squared, heritability is h squared, and in this case, both of those things are 0.9165. So don't let that confuse you. You don't have to do any numerical machinations with that. So what we see is we can use this figure that I've got on the screen for you here, such that the selection that is acting on the parental population multiplied times the heritability of the trait that we're measuring, those two things multiplied together will predict what the evolutionary response to selection is. This is the breeder's equation, and here it is formalized for you. So the heritability value is 0.9165. Once again, you don't need to do anything to it. In this case, the figure before, if you actually had the data available to you, you would see that the parental generation shifted by 2.41 inches from the pre-selection average to the post-selection average. And when you multiply those two things together, you would expect a shift in the expected offspring phenotype to be 2.2 inches. Now, the reason that you had selection on the parents of 2.4 inches of increase, but you only observed 2.2 inches of increase in the offspring is because heritability is not perfect. If heritability was perfect, you would find that the heritability estimate would be 1, and then the shift in the parents would be exactly the shift in the offspring. So one thing that you should know about heritability is it has the theoretical boundary at 0, where the trait is not heritable at all, and an upper boundary of 1, where the phenotype of the offspring is determined entirely by the phenotype of the parents. So go back to this figure, and a slope of 1 is essentially equivalent to the slope of this red line. And if this red line continued through the end of this graph, any selection on the parents would not lead to any change in the offspring because that slope is 0. And I think I said a minute ago that was a slope of 1. It's not a slope of 1, it's a slope of 0. And if you took this line and canted it upward just a little bit, you would be getting closer and closer to a slope of 1. As you cant that line upward, you would see that the evolutionary response would also widen because the slope is higher. So, heritability goes from a low estimate of 0 to a high maximum of 1. And this is a pretty high estimate for heritability. This is high even for estimates of heritability of height in humans, which is already a characteristic that is kind of on the high side of heritability. So this is how the breeder's equation works. Now imagine that you are a dairy farmer and you want your cows to produce more milk. You could look at your cattle herd that you have, and you could actually look at the mothers and their offspring, and you could basically measure milk production in the mothers, milk production in the offspring, and you could calculate this heritability. And then you could use that and say, okay, well, if I want my cows to go from each cow producing 12 gallons of milk a day to produce 14 gallons of milk a day, you could put that change of 2 gallons of milk in the evolutionary response to selection area. You know the heritability because you measured that on the herd, and then you could solve for selection, and that would tell you how much you need to actively select who gets to breed in your herd and who doesn't get to breed in your herd. So that's why it's called the breeder's equation. It was originally intended for agriculturalists to know how to breed their animals to get a desired outcome. So in this, if you know two of these things, you can always figure out the third. If you know the evolutionary response to selection and the strength of selection, you can figure out heritability. And indeed, there are some systems where it's much easier to actually exert a certain amount of selection on the population, observe the response to selection, and calculate heritability that way. In other systems, it's easier to measure the phenotype in offspring and the phenotype in parents and do a parent-offspring regression. But if you know any two of these things in this equation, you can always figure out the third. Once you've gotten rid of that population, though, now the population has less genetic diversity because the only individuals that are hanging out are a bunch of individuals who had a predominance of dominant alleles. And when you calculate a regression on this reduced cloud of points, you see that now the slope of that line is considerably lower. It was 0.91, now it's down at 0.66. And so when we think about directional selection, and this is one of three types of selection that we're about to go through, one of the things about directional selection is it has two effects on the population. One is it shifts the average phenotype value. It'll either make the average phenotype larger in value or smaller in value. But the other thing that it does is it reduces the additive genetic variance. It reduces the genetic variation that is in the population. And we can actually quantify or identify different forms of selection by paying attention to how that form of selection affects the average phenotypic value and what it does to the variance of the phenotype. So as we just talked about, directional selection is when you select against one extreme of the distribution or the other extreme of the distribution. Don't lose sight of the fact that these bars in this distribution graph are all due to the presence of alleles that are inherited by standard Mendelian mechanisms. But when you select against, in this case, the lower portion of this distribution, we have the pre-selection distribution here that is a dotted line. We have the post-selection distribution, which is the solid line. And we can see that the mean has shifted, and that's the evolutionary response to selection. But not only that, the variation in the pre-selection population is higher than the variation in the post-selection population. And so not only has the mean shifted, but the variance of the population has also shifted. And this is the hallmark of directional selection. So on the last test, when we were talking about how selection in a general way can lead to either increases in genetic variation or decreases in genetic variation, and it depends on the type of selection, this is what we're talking about. In this case, directional selection leads to a decrease. Stabilizing selection is a different form of natural selection, and in this case, both extremes of the distribution are selected against, which means that you're only left with this intermediate set of phenotypes. The result of that, assuming that the selection is perfectly symmetrical, is in theory, once again with perfectly symmetrical selection, the average phenotype value doesn't vary between the pre-selection distribution, which you see is once again a dotted line, and the post-selection distribution, which is a solid line. Now, everything has to be perfect for it to not shift, but even if it's somewhat asymmetrical, it's going to not shift very much. But in this case, once again, because you're removing these extreme phenotypes at the edges, the variation in the post-selection population is lower than the variation in the pre-selection population. So stabilizing selection could be defined as a type of selection that results in little to no change in the phenotypic mean and a decrease in the phenotypic variance. And the last form of selection is disruptive selection. And this is the type of selection where it basically selects, the environment selects against intermediate phenotypes. And what that does is it's like driving a wedge down the middle of the distribution, and what you get in the next generation and future generations is this bimodal distribution. Now, once again, assuming that that disruptive selection, removing intermediate phenotypes from the population, is completely symmetrical, once again, the average phenotype doesn't change. But what happens is future generations, more of the individuals in the population are distributed out towards the tails. Fewer of them are clustered around the mean, as in the pre-selection population. And what that does is as you move the density of that distribution away from the mean, the variance goes up. And so disruptive selection could once again be defined by what happens to the mean and what happens to the variance. In this case, the mean doesn't change and the variance actually increases. So my tendency is to think of these three forms of selection in terms of effects on the mean, and effects on the variance. And that's more than likely how I'm going to ask you these things on an exam. Now, when we look at something like selection in Darwin's finches, I mentioned this early on when we were introducing evolution, but this is a case study from back in the 1970s. These researchers had been following Darwin's finches, looking at what they ate and how the beak of the finch was evolving over time. And they happened to be there during the period from 1975 to 1978. In 1975, the islands were subject to an El Nino. Normally, the Galapagos Islands are in the middle of a cold ocean current. Because the ocean is cold, you don't get a lot of evaporation off of the ocean, and so these are literally desert islands. So whenever you are thinking about what your musical desert island mix would be, you can think of what music would I want to listen to when I'm on the Galapagos Islands. But when there's an El Nino, the islands are surrounded by warm water. You get a lot more evaporation, and as a result, you get a lot more rainfall. So in 1975, there was more rainfall. More rainfall led to there being more plants produced more seeds were eaten by birds, they reproduced really well, and the population was really high up here at around 1,400 birds. But when the El Nino went away and the cold waters came back, the rainfall declined, and the only food that was available to the birds, because the plants all died, were the seeds that were left from all the abundant plants that were there before. And because there were so many birds, they ate them. And as they ate them, you can see in this next chart how the abundance of seeds is going down, because these 1,400 birds are eating the seeds, and as seed abundance goes down, a lot of these birds are actually dying until you get to the end of 1977, and the population size is plummeted from 1,400 birds all the way down to 200 birds. So that's a really big population die-off. That's a severe selective event. Well, this lower left-hand figure down here is showing you an index of seed hardness. So some seeds are big, some seeds are small, depends on the species of plant that the seed is associated with, that it comes from. And you see that here at the beginning of this graph, seeds are of intermediate size. And you can see that over time, the seed size and hardness goes down because they're eating the intermediate seeds, then they're eating the small seeds. And once they've eaten all the small and intermediate seeds, the hardness of the seeds goes way up. And so by the beginning of 1977, when the population is down to about 600 birds, the only seeds that remain on the island are the very hardest seeds, which means only birds with the biggest, deepest, strongest beaks can survive. And as a result, here, this last figure in the lower right-hand side here is the overall size of the beak. And it starts off at a relatively small size, and you can see at about the same time that hard seeds are the only seeds in the environment in 1977, all of a sudden the size of the beaks of the finches begins to increase, and by 1978 it's about 80% deeper and larger than it was in 1975. This is what directional selection does. It shifts the average phenotypic value from one value to another value. In this case, because the food changed, because there was an environmental event that affected the availability of food for these finches. Directional selection. When we look at the parent-offspring regression of the 1976 finches and the 1978 finches, it's interesting. The red dots and the red line here represent families in 1976, and this cross here represents what we call the bivariate mean. It's the average value of the parents versus the average value of the offspring. And one of the things that you'll notice is that the phenotypic range in 1976 goes all the way from a mid-parent build depth of 8 up to a mid-parent build depth of 11. And you'll also notice that the 1976 line, this red line, is lower in terms of its y-intercept than the 1978 line, which is the blue line. When we look at the blue line and the blue dots, you'll notice that the bivariate mean is higher. The bivariate mean from 76 was about 9.5. By 1978, it's almost up to 10 for the parents, and the bivariate mean in the offspring is also shifted upward. But what I want you to also pay attention to is look at the range over which the blue dots span, and you can see that there is a lack of finches who have mid-parent build depths in the 8 to 9 millimeter range. And so once again, this shows you that directional selection has resulted in an increase in build depth in the population, and it has done this by basically getting rid of small build finches. When we think about disruptive selection, a good example of this is the Illinois corn oil experiment. This is a group of researchers in Illinois who have been growing corn plants for 100 generations of corn. And what they did was they started off with corn oil content in the seeds of the plants that was about 5% of seed mass. And what they did was they took their corn crop and they divided it into two populations. In one population, they would only allow corn plants that were producing more than average oil in their seeds to reproduce and form the basis of the next generation. That's the continuous high oil population. Then there's a continuous low oil population where they only allow those plants that are producing lower than average corn oil in terms of content in the seeds. They only allow those low corn oil plants to make up the next generation. So we also have selection for continuous low oil. And what you can see is that when you're selecting for high oil content, the population increases in oil content, and it increases at a pretty good clip. But when you look at the continuous low oil line, you see that the oil content is going down, but it's going down at a much slower rate than the continuous high oil content is going up. At about 48 generations in, they reversed selection, so they divided the high oil selection lines and continued some high oil selection lines, but then they also reversed selection on those high oil lines where they started selecting for low oil. And once again, you see there was a very quick response to that. And they also did the same thing in the low oil lines, except they reversed it in this case to produce higher oil. And you can see that the population responds, but it's responding at a much slower rate. About 10 generations after that, the reversed high oil lines, they re-reversed, and they continued some of the plants on the selection for low oil, but then re-reversed it so that they now have a second line that is being selected for high oil. And you see that both of those lines respond to that selection really well. But when we get out to around 90 generations, the continuous high oil population here in yellow is beginning to converge with the reversed high oil line up here. And so what I want you to stop and think about for a minute is, Why does the high oil content line respond really well to selection, but the low oil content line does not? And why, at the end of this time frame over all of the selection, do these high oil content lines begin to converge on one another? So I want you to pause the video for a minute, and I want you to think about that and come up with some ideas as to why this pattern of the speed of evolution is different in these two different lineages, and why those high oil selected lines are beginning to converge with one another. So pause, think about it, write down what your thoughts are, and then start the video when you're done. Okay, presumably what's happening here is oil content in corn is due to quantitative genetic variation, additive trait variation. And what you have is at 5% oil, you probably don't have very many of the alleles that contribute to oil production in the kernels of corn. And as a result, there's not a lot of genetic variation for low oil content because most of the alleles are already alleles that result in low oil content. So selection is acting on a population that doesn't have much of that type of variation. Because you have a bunch of lines that are at 5% oil content, any line that is producing more than average oil content will tend to have more dominant alleles, presumably, in that in those individuals, so that when you select, you're selecting for those alleles, and there's just a lot more room in the genotype to add dominant alleles than there are to remove recessive alleles. So as a result, the high oil content line responds quickly to selection. The low oil content line responds poorly to selection or slowly to selection. When we get up here to these two high oil lines, the continuous high oil that's been selected on for 100 generations and the re-reversed high oil, you see that they begin to converge on one another up at around 20% oil content. Well, presumably now the same thing is happening at the high oil end of the spectrum, where in this case, all of these individuals have a lot of dominant alleles that contribute to oil content, and as a result, they're beginning to lose genetic variation for oil content. They're just losing genetic variation at the high end versus losing genetic variation at the low end. I'm going to go back and pull up a slide from when we were talking about the four forces of evolution, or the five forces of evolutionary change. And I want you to remind yourselves of what genetic drift does to populations. Remember, I showed you a series of simulations of populations drifting around as a result of random genetic drift. And the zero mark down here on this graph and the 1 mark over here is basically the frequency of the dominant allele. And populations are drifting either towards eventually being fixed at 1, where there's nothing but homozygous dominance, or they're drifting around until they're fixed at 0, which is a population of nothing but homozygous recessives. And as you can see, these graphs, as they drift around, especially this one at a population size of 8, look similar to the kinds of graphs that you're seeing in the corn oil experiment, where you're seeing them basically, they're not drifting upward, but they are being propelled upward by natural selection in a consistent manner until you begin to get to fixation for all loci that might contribute to high oil content. And these are not drifting downward towards 0, but they are being selected downward towards 0 until here by about generation 80, all of the plants are basically at 0 oil content, and that would represent a complete loss of genetic variation in the other direction. So with that concludes kind of our coverage of genetics. And what I want to do next is go in and start thinking about applications of natural selection and the influence that natural selection has on speciation. So here at the beginning of this particular slide deck, you're going to see that we look at some learning goals. So one of the learning goals is to understand how natural selection can be applied to artificial selection, so selective breeding of plants and animals. And then I also want us to learn in this section how different genetic mechanisms can lead to similar phenotypic and fitness outcomes. A good example of one way to think about this is there might be multiple paths to avoiding predation, and because those multiple paths probably involve different genes, you can avoid predation one way or another way, and depending on which way your population responds to predation, that will maybe lead to similar outcomes, even though there's a different genetic path to get to that same outcome. And then the last thing that we'll talk about is understanding how natural selection leads to the origin of new species. And this will increase biodiversity. So we've been talking about central dogma and talking about how phenotypic variation comes into being at a very molecular level and how that influences phenotype. We've talked about the heritability of phenotypic variation, so we spent a lot of time in genetics. Now we're very much in the part of the course where we're talking about natural selection and how phenotypic variation affects reproductive success and survival. We just went through the three forms of selection, so I'm going to go very quickly through this because these are all just new examples of the three forms of selection that we've already talked about. This first one here is disruptive selection leading to two peaks. This is, once again, the type of thing that happens in Darwin's finches. This is how you had the radiation of Darwin's finches into small-beaked finches, large-beaked finches. We also have directional selection. This is an example with selection for negative phototropism. Phototropism is just the tendency of an organism to want to move towards the light. This is easy to measure in animals. And negative phototropism is your tendency to want to move away from light. And as they selected for negative phototropism, you can see that the tendency to fly towards light goes down. And this is only over the course of about 20 generations. But once again, when you get to about generation 14, evolution has kind of leveled out and stopped moving, presumably because you've exhausted genetic variation for this particular trait. And then the last form was, of course, stabilizing selection. A good example of this is birth weight in humans. And so we have the red line here represents infant mortality. Premature babies, this is some data from these two authors, Carn and Penrose. It was published in 1951. It's a pretty famous data set. It's called the Carn and Penrose data. The premature babies in 1951 don't survive very well, and babies that are too big in 1951 don't survive very well. So the ones that have the best chance of survival are these babies that are clustered around the average birth weight, and this results in a distribution that is relatively narrow, but once again has its main mode at the kind of average birth weight, and this is characteristic of what you would expect under stabilizing selection. Nowadays, we have much better postnatal care for premature babies. So oftentimes hospitals will have pediatric units that are designed to care for premature babies, and so there are premature baby ICU units. And so this mortality line for premature babies has gone down. It's not down to normal weight babies, but it's considerably lower than it was. And because we do cesarean sections whenever there's a complication with a pregnancy due to the size of the baby, we don't go through a natural birth. We just remove the baby via cesarean section. And so selection at the two tails here, in modern times, is probably being reduced, which means that you should probably expect this phenotypic size range of babies to broaden out over time as we use medical interventions such that we've reduced the overall mortality rate. We've basically reduced the strength of stabilizing selection. So we're still talking about quantitative traits. We just went through this earlier in this screencast, so once again, I'm not going to belabor the point, but these heritability figures are still in here. And then we have disruptive selection, which we talked about with the corn oil experiment. So this is all now in the realm of review. So if you look at this corn oil experiment, it looks like lineages in a phylogeny. You have a common ancestor that is branching into a continuous low oil line and a continuous high oil line, and then that splits when they reverse the oil content selection that they're doing. And it's this divergence that was actually impressive to Darwin. And back in 1837, he opened up his notebook and drew this image that you see on the left here in his notebook, which shows an ancestral species here at 1 that branches repeatedly until you have species A, species B, species C, and species D. And then you have a bunch of other unlabeled species that represent species that have gone extinct. Here's the figure that finally ended up in Darwin's book, The Origin of Species. And you can see that there are extant species here at the top, and you see some species and some lineages that just basically went extinct over time, and these are the ones that ultimately survived. And so Darwin was the first person who came up with this idea that populations get split by natural selection into 2 subpopulations that then go off on their own evolutionary trajectories. Here's a very modern version. This is a phylogeny of 3,000 species that include everything from archaea to bacteria to protozoans to plants to fungi and animals. Human beings are right here on this figure. This is a circular phylogeny that is basically designed to get at the unity of life. And so this is how we envision evolutionary phylogenetic trees nowadays. But you can see here that there's a common ancestor that gave rise to all of the fungi. Here there is a common ancestor that gave rise to all of the animals. Here there is a common ancestor that gave rise to all of the plants, etc., etc. So one of the things that we want to think about is whether or not there are limits to evolutionary change and what those limits might be. And if you watch nature documentaries, you will hear the narrators talk about how natural selection has the ability to shape organisms to be perfectly adapted to their environments. And while natural selection can be really good at that, and it can be deceptively magical at how well organisms are adapted to their environments, that's a little bit of an oversimplification because things are always more complicated than that. If you look closely at almost any of those sort of textbook or documentary pronouncements and start to look closely at those cases, you will see that those organisms are actually less perfectly adapted to their environment than they could be. And this is because evolution's goal isn't perfection. Evolution's goal really is just adequacy. Can you survive better than the other individuals in your population? That's what gets you ahead evolutionarily. And along the same vein of thought, there are actually some things about genetics that really can stand in the way of evolutionary change, moving a population towards some pinnacle of ultimate fitness. And these are pleiotropy, a loss of genetic variation, and interactions among genes. So we haven't used this term pleiotropy yet, but pleiotropy is a situation where you have a single gene that controls more than one trait. An example from your textbook is comb size and egg production in chickens. The comb is that fleshy appendage on the top of the head of a chicken. And egg production, of course, is all about producing eggs, but eggs are surrounded by a shell that is made up of calcium. And it turns out that there are genes that affect bone density, and the genes that affect bone density are actually genes that affect the expression of cartilage and the development of cartilage. And so when you start making a bone, the first stage of bone production is to create cartilage, and then that cartilage becomes impregnated with calcium carbonate. So genes that control cartilage formation control bone density, but the genes that control cartilage formation also control comb formation because combs are made up of cartilage that doesn't ever harden into bone. So you have a single set of genes or a single gene that controls both the density of bones and the size of combs. And turns out that the bone density in a chicken influences how many eggs they can produce because they mobilize calcium away from their bones into producing eggs. And so all three of these traits ended up being correlated because they're all controlled under the underlying control is from a set of genes that are basically all about controlling how cartilage develops. Therefore, if you select for higher egg production, you're also going to be selecting for a bunch of other traits, and that selection on egg production will drag along those other traits, such that if it's better to have lower density bones because it makes you lighter and able to fly, that is in conflict with selection acting on the production of more eggs. And so that can actually slow down selection of those two traits. When we think about a loss of genetic variation, we've already mentioned it with a corn oil experiment, but this is a figure from your book looking at racehorses. And this is the finishing times of racehorses in the Kentucky Derby. And you can see that back in the late 1800s, racehorses were very slow relative to modern racehorses. But then breeders were breeding racehorses to be faster and faster. And with that selection, you saw that the finishing times in the Kentucky Derby actually went down at a pretty fast clip. There's a really fast decrease in the finishing times, meaning faster and faster horses. And by about 1980, you don't see much change in the finishing time of horses at all. And that's because they've probably exhausted most of the genetic variation that is responsible for fast horses. And these variations that you see down here are probably more environmental effects like quality of the jockey, quality of the training regime, condition of the track on a particular day, and things like that. So you can also have evolution stop simply because you don't have any more genetic variation for whatever that trait is. And then the last of these, and this is where we'll finish up, is gene interactions. So this is a study done by a researcher at the University of Virginia. He was in Oregon studying a population of snakes and discovered that this population of snakes had a striped morph and a colored morph, a checkered morph rather. These two snake morphs are found on the same plot of land. They're in the same species, Thamnophis ordinoides, and he was interested in the relationship between color pattern and behavior. Obviously, color pattern and behavior are probably determined by different genes. So he was racing these snakes down racetracks, and as they were racing these snakes down racetracks, they noticed that some snakes would just take off down the track in a straight line, and they would go all the way to the end of the racetrack without any problems. But other snakes would get partway down the racetrack, and then they would turn around, go the other direction, and freeze. And this was frustrating to them because they wanted to race the snake to the end of the track. But it was frustrating enough to them that they were paying attention to which snakes did this reversal behavior and which snakes didn't. And it turns out that striped snakes tended to slither in a straight line, whereas checkered snakes tended to reverse themselves and freeze. And so this researcher, a guy named Butch Brodie, began to think, oh, maybe these two characteristics, the color pattern and the behavior, are correlated with one another, not because it's a pleiotropic effect, but because these two genes become correlated with one another because of the influence of selection. And so what they did was they scored the snakes that they were racing for color pattern. And so you have some snakes with a high stripe index, those are the stripy snakes. The low stripe index, those are checkered snakes. And as they were racing them, they also scored the tendency to reverse themselves. Individuals with a high reversal index tend to reverse themselves. Ones with a low reversal index tend to go in a straight line. And then they released these snakes into the wild, and these snakes were collected at a hibernaculum, a place where snakes go in the winter to hang out with one another. And then in the springtime, they go out into the fields and pastures and forests. And then in the fall, they come back to that same hibernaculum where they were the previous spring. So they marked these snakes after they raced them and scored them for behaviors. They released them into the field, and then they collected everybody who came back to the hibernaculum, and they looked at who returned and who didn't, and they made the assumption that if you didn't return, it was because you were probably killed and eaten. And what they found was that striped snakes that went in a straight line, had a low reversal index, survived better than anybody else. Checkered snakes that reversed themselves were the second best at surviving. Checkered snakes that go in a straight line were third best, and striped snakes that reverse themselves were really terrible at surviving. So the selective environment was such that being striped or checkered had an adaptive value, but only depending upon which set of behaviors it was paired with. And this goes back to peculiarities of the visual system of vertebrates. But basically, being striped is fine, but only if you slither in a straight line. Being checkered is fine, but only if you reverse yourself and freeze and become cryptic. If you're checkered and go in a straight line, you tend to get killed more often. And if you are striped and you reverse yourself and freeze, you tend to get killed more often, because it's really easy to see a striped snake laying still on the forest floor. And so these two genes, the genes that control color pattern and the genes that control behavior, are correlated, not because they're on the same chromosome, which that's one way to get correlated evolution, is because you have physical linkage on the chromosome. They're not correlated because they're coding for functionally related characteristics like the comb and egg production in chickens. These two genes become correlated with one another because they're in a selective environment that makes incompatible combinations of genes be selected against, and only compatible versions of these two traits are allowed to survive. So we will start at the beginning of class on Wednesday with looking at Trinidadian guppies and how they evolve in response to selection. So no lab tomorrow, Wednesday, but we will have class on Zoom. Thanks for listening, and come to class tomorrow with any questions you have.